everyone, and welcome to today's webinar, which is being hosted today by Dicel Arbor Biosciences. The session will be recorded and made available online for later viewing. My name is Allison DeVault, and I'm the Director of Genomics at Dicel Arbor, and I'll be moderating today's webinar. Our topic today is advances in resistance gene sequencing, or RenSeq, for crops via targeted NGS. This is a two-part series. This is the second part. The first part was held in February, and the link to that recording is available on Arbor's website for anybody who missed it. This workshop was originally intended to be held at the PAG conference in San Diego this January. That was unfortunately canceled at the last minute due to Omicron. We're very grateful that all of our speakers were able to switch to this webinar format at the last minute. So before I get into the science, I just wanted to let everybody know that we will accept questions from the audience today for our speakers via the questions portal in the webinar control panel. You can submit a question at any time throughout the session, and it will pop up for us, and we'll go through those questions at the end of each speaker's talk. So please do feel free to submit them at any time, and our apologies in advance if we don't have time to get to yours in the live session today. So before I hand things over to our speakers, I'll just quickly review some concepts that are important today about RenSeq. So one of the most agriculturally important groups of plant genes are known as the R genes. This is a complex family of genes which typically encode for nibbler or NLR proteins. In plants, these genes function in disease resistance for many pathogens such as fungi, bacteria, viruses, and more. Of course, this makes them incredibly relevant to understand and develop in an agricultural context. RenSeq is the technique of resistance gene enrichment sequencing, which utilizes targeted next generation sequencing via hybridization capture to enrich novel and unknown R genes sequences from plant NGS libraries built from DNA or RNA. These are then sequenced on long or short read NGS platforms, and then can be the enriched sequences can then be digitally reconstructed with bioinformatic analysis. This is a powerful approach which can be deployed in various programs for discovery, trait improvement, and more. At Dicel Arbor Biosciences, our myBaits line of target enrichment kits can provide custom and pre-designed hype capture that can be used in the RenSeq enrichment workflow. And with our myReads, which is our full service NGS laboratory, our team of experts can perform any and all of these steps for the whole NGS workflow used in RenSeq. Many different studies on many different species for both plants and pathogens have utilized myBaits kits and or our myRead services for their studies in RenSeq, including wheat, potato, Arabidopsis, and more. I'm highlighting just a few of those publications here, but there are quite a few more that are linked on our website, and you can also find additional information there about RenSeq, as well as our contact info where you can reach out and contact us if you would like to discuss a project idea with our team. For today's webinar, we'll be featuring talks by two researchers who are working in cutting-edge plant genomics. Our first speaker will be Sanu Arora, who is a group leader at the John Innes Center in the UK. Dr. Arora currently investigates the genetic basis of disease resistance in peas, the goal of improving crop yields and stability. Our second speaker will be Ingo Hein, who is an associate leader of the potato group at the James Hutton Institute and reader at the University of Dundee in the UK. Dr. Hein is currently studying potato disease resistance genetics and pathology with the goal of increasing resistance to pathogens. So today, Dr. Arora will be presenting first on her work on the genetic basis of pathogen disease resistance in peas using the RenSeq approach. So before I hand things over to Dr. Arora, as a quick reminder, if you would like to ask a question while the webinar is running, please feel free to use that questions function in your webinar control panel, and we'll read through those questions after the two speakers talk. So with that, give me just a moment and I will transfer control over to Dr. Arora to give her presentation. Thank you very much. Looks good, thank you.
Can you see my screen?
Yep, we see your screen and we hear you just fine.
Okay, so is it full screen presentation mode?
Yep, looks good.
Okay. Thanks, Alison. Hello, everyone. I am Sanu Arora, group leader at the Downy Center. The focus of my research program is to recruit genetic diversity from wild relatives and land races to develop resilient crop varieties. I've recently started my group to investigate pea disease resistance. Legumes, including peas, are the essential components of the sustainable agriculture. They contribute to soil fertility by fixing atmospheric nitrogen and promoting soil microbiome diversity, which in turn lead to reduction in the use of fertilizers in the soil. They are also an excellent source of plant protein, and they substitute for ruminant-based diets, so they also contribute to the health and as well as to the environment. In short, legumes are critical to achieve carbon neutrality by reducing net greenhouse gas emissions. So its legumes are the crops that fall into the category which benefits the environment and human health and has a lot of other benefits. It is encouraging sign to see that in recent years there has been a noticeable dietary shift from meat to vegetarian products, leading to an increase in demand for pea protein, which is expected to grow exponentially in the coming years. So this is 2023, but it's expected that by 2026 we will have even more demand than what I've shown here. However, there is a highly volatile yield of the peas that is observed at the farmer's field. So in some years, the crop behaves very well, you get a good yield, and sometimes there's disease pressure and other reasons where you will get a very volatile yield, you'll get a low yield in another year. So what is interesting at this point is, can we cope up with the demand that is exponentially increases, but it looks like with the volatile yield, it is a difficult scenario. So the important question is, we need to understand why there is this volatile yield that we see in farmers' field. So there are multiple factors causing this yield volatility, such as biotic, abiotic stresses, and subpar agronomic potential. Understanding the genetic basis of these causes will help us to achieve the holy grail of yield stability. My focus is particularly on identifying genetic sources of resistance for some of the important diseases of peas against which the current control strategies, which is in use of the chemicals, are not particularly effective or not available. So pests and diseases are the two most important biotic constraints affecting global pea production, causing yield losses that can range from a small percentage to complete crop loss. Despite our increased use of pesticides and other polluting chemicals, we are still in a state where our crops suffer huge losses each year due to these biotic stresses. Is it possible to protect our crops without harming the nature? And the answer lies with identification of novel sources of disease resistance as a first step towards breeding for resistant cultivars. And one of the focus of my research program at the Downy Center is to understand and explore the genetic architecture of disease resistance in Pisum species and work with the breeders closely and agronomists to translate the outcomes of this research into the breeding programs. Here is a list of some of the important diseases of peas. So I've put this picture for some of you who might be new to the pea diseases. It mostly includes mildews, powdery mildew, downy mildew, root rots, which is one of the devastating diseases of peas, in the field. And some other include blights, aphids, weevils, and rust. So occurrence of these diseases depends upon their geographic location, climatic conditions, and soil health. So to find new sources of resistance, I focus on going back to the crop wild relatives and land races to find new sources and transfer it into elite cultivars. Just a couple of years ago, cloning a single gene from wild relatives by crossing into cultivars was a daunting task, and it would take several years to clone a single gene, followed by a number of years that it will take for the integration into the varieties. A case in point, there has only been one gene mapped so far for downy mildew resistance in pea, and it took over a decade to map this gene and introduce into elite cultivars. And when it was deployed in the field, this resistance was overcome within a few years. So it took more time to discover the gene than actually for the pathogen to overcome. So examples like this presses the need for a robust program of finding new genes and deploying them in stacks, either through transgenics or the breeding programs. With the recent breakthroughs in sequencing technologies, particularly in the terms of cost reduction and development of novel genomic tools, that we can now rapidly clone multiple resistance genes in a reasonable time frame and deploy them in stacks to achieve resistance, which is considerably more durable in field conditions. One such method is using RenSeq in combination with association genetics. I developed this method in my previous research, and we dubbed this method as RenSeq. Here is the flow chart of this method. We phenotype a crop-wide diversity panel with a pathogen isolate, and at the same time, we enrich the genomic fragments of each accession using an NLR-based library. Most of the gene clones for resistance, as told by Allison, they are reported to be NLRs. So for the complexity reduction, we only sequence the NLR contents using RenSeq approach. When both the sequence and the phenotype data are available, we use association genetics to uncover new sources of resistance. For the first time, I implemented this method in the Aegilops tauschii panel. Aegilops tauschii is also called goat grass, and it is the genome progenitor of bread wheat. We have about 200 lines in the panel, which are divided into 3 genetic lineages indicated by red, blue, and green colors here on the tree. We took lineage 2, which is the red one, which has been reported to have more sources of rust resistance. And we phenotype the panel with the 3 races that I've shown here in the boxes for the stem rust, RKQQC, TPMKC, and QTHJC. So stem rust is an important disease of wheat. And the outcome of implementing RenSeq on this panel and the set of phenotypes was these 3 graphs. We got 4 exciting peaks indicated with the red color. The one on the first one, SR33, was one of the positive control, a gene that was already known to be found in tauschii and was cloned. And another gene, SR45, that was also a gene that was known by the time we did the work. So, but we found 2 exciting candidates. So one is SR46 and another is on the bottom graph, SRTA1662. So these contigs, so these peaks, they underlie single contig. That's the beauty of RenSeq, that you get sometimes a single peak which underlies a single contig. As for example, in the case of the last graph with the QTHJC, we got this peak, red peak, which underlies a contig of about 10 kb in length, which had, when we annotated, it had CC and BARC NLR domains, a typical NLR gene structure, and we had to do some lab work to understand the complete structure of this gene. And following that, when we had the complete gene ready, we introduced it into a susceptible wheat variety called Fielder. So Fielder was susceptible to this race QTHJC. When the gene was introduced into this variety, we saw resistance in these lines, which is clear from the transgenic picture that I have. The one with single copy, these are far more resistant lines compared to the nulls, which show complete susceptibility. So it was the use of this single gene, transformation of this single gene into the Fielder was enough to provide the resistance. So this is a beautiful example of using RenSeq method for gene cloning. Recently, when I started my program on finding diverse sources of disease resistance in the Pisum species, so the exciting thing was we had this fantastic pea germplasm collection on which I decided that we would implement the RenSeq approach. This work was started with Claire Domini's lab at the John Ennis Center. To introduce the P collection at JIC, we have about 3,000 lines in this collection. And based on genotyping with retrotransposome-based markers, the collection was divided into 3 main subgroups corresponding to land races, cultivars, and Pisum, wild Pisum lines. So it's like a rough division. When I say mostly land races, there are some lines which will be cultivars and a mix of others, but mostly this division was good enough. So the white group on the far right, that's very interesting. It constitutes a little part, but it's a very interesting feature here. So the wild species, there are different subspecies. There are different species of Pisum, which includes Pisum epsinicum, Pisum elateus, Pisum fulvum, and humile. So, and this little one has a lot of unexplored diversity in it. It is in the recent study, it is reported that 50% of the alleles were shared among wild, landraces, and cultivars, while 25% were unique to wild accessions and 5% in landraces. So this data indicates that Pisum is a diverse species and we could benefit from integration from its wild relatives. So this is a picture showing incredible diversity of this panel. I had humbling experience growing them in the glass houses at GIC last summer, and there is a beautiful diversity, that phenotypic diversity that you see when you're growing the panel, diversity for pod, shape, size, flower color, plant height, response to diseases, and a lot more. For RenSeq, we selected about 230 lines based on the genetic diversity of this panel, and also we used some historical data for the disease phenotype. The top bar in this slide represent the three groups that I mentioned before, which are further subdivided into the genetic groups indicated by 1.1, 1.2 to further to 3.6. We overlaid the genetic subgroups with disease phenotype data in the third bar, and then we made a selection of about 230 lines that cover both the genetic and disease variation. These lines were sequenced for their NLRs with RenSeq done by Arbor Biosciences. Here is an overview of the distribution of NLRs on the 7 chromosomes of P. So this is analysis done on one of the P reference genome, Chemior. So this is all the 7 chromosomes have NLRs distributed across, except for the short arm of 3, chromosome 3 and 4, which look a bit sparse. So these are about 185 NLRs from the Chemior genome. And we used different resources for designing the bait library, and most of it, when 66% of the resources were Pisum genomic resources, which included about 8 sequence lines, and others were designed from closely related species like Vicia faba, Medicago, and Lens culinaris. So in total, there were about 40,000 baits of 120 base pair size. We implemented RenSeq pipeline in P to find genetic loci involved in downy mildew resistance. Downy mildew is an important disease of pea, causing about 40 to 75% yield reduction and has been a problem since the 1960s. It compromises growth of the plant and later spread to pods. It causes, the causal agent of this disease is Brenospora vicia, subspecies PC, and it is an oomycete which survives in the soil even after long rotations, which ultimately impacts the future crops as well. So the primary control is used through the use of the seed treatment with some fungicide. One of them I mentioned here, Vocil. But some of the fungicide has been recently restricted for their use in agriculture, further pressing the need to find genetic sources of resistance. So when we got the RenSeq data, on the first downy mildew data set that I used was actually the historical downy mildew data that was actually also used for the selection of these lines. The graph at the bottom shows the phenotype variation for this data. So I implemented RenSeq pipeline on this data, and a clear peak was seen on chromosome 2. This peak underlies a couple of NLRs, which are in linkage disequilibrium. Looking at the distribution of this P in the 230 lines on the right-hand side, so this is a phylogenetic tree of the lines sequenced with RenSeq and the tree made using that data. So you can see in this tree, the red and the blue color indicates the phenotype data, resistant and susceptible lines. The resistant loci, I was interested to see how many lines carry this resistant loci, which we found on chromosome 2, and only 8% of the lines carry this loci, which I've indicated on the outer circle with black color filled dots. And those lines which are unfilled doesn't have this loci. So interestingly, this, as you can see, this loci is present on one side of the tree. There's other side of the tree which doesn't have this gene present. But this gene only explained 8% of the resistance in the panel. So there was a lot of unexplained resistance still there. So I was, when I started with the historical data in RenSeq, I was interested to know what is the diversity of this pathogen. So there is a report that was prepared by PGRO in 2018. PGRO is Processors and Growers Organization in the UK. They surveyed pea downy mildew races within UK, and based on their report, there were at least 15 different races identified. 5 of these were new races indicated with UNK12, unique 125, or unknowns. And 5 of these unknowns, and while the other 10 races have been already found in 1980s. So this race structure in the study, this indicates that the races, new races, they evolve and evolve quickly, and the pathogen evolves, and we need new sources of resistance and prepare our crop for the evolving pathogens. So what we did, we chose 2 races from this set of races that I mentioned here, and we phenotyped the panel in collaboration with Jane Thomas and Tom Wood at NIAB in Cambridge. And this is the scale of phenotyping that we used to observe the variation. And you can see the disease survey was done at the seedling stage. When they were inoculated with two different races, it was done at 2 different time points. Though these 2 races, when we picked, we picked them based on the differential set, and there was some differences, and they were also collected over different years and from different location. But the data from the 2 races, it looked highly correlated. The interesting aspect was some lines, it showed complete resistance. So I have marked here like resistance. So there were some lines in the panel which were completely resistant. So that was really interesting, and some lines which were completely susceptible, going to 80 to 90% susceptibility. But anyways, both the races behave very similar in the terms of phenotype data set. So when we did the analysis with both the races separately, but we got the same peak on chromosome 5 with both the races, which was expected because the data in the end was highly correlated. And this is a peak you see on chromosome 5, a very clear peak. And when I analyzed the distribution of this resistant loci, so out of 120 resistant lines indicated with the blue color in the phylogenetic tree on the right, 48 carried this loci. And again, the same observation was in this case, the resistant lines carrying this loci was only present on one side of the tree. There's another side of the tree where we couldn't see both of the loci that we identified. So this was an interesting thing. There was about 48 lines out of 120, which was, we could see there is still unexplained resistance. So we are doing further work on this resistant loci. We're designing some gene-based markers and making crosses to transfer this resistance to the elite cultivars, and also doing functional validation of these both these loci. Now, as there was a lot of unexplained resistance in the panel, what we did was we decided to remove all the lines which were black filled circles, basically containing chromosome 5 resistant loci, and rerun the analysis. And then what we saw was another peak on chromosome 1 and chromosome 2. The chromosome 2 peak is the same as we found with the previous historical isolate of downy mildew. So this resistant loci works against both the races, and there's another peak on chromosome 1 which we are exploring currently. So to add up here, so there are different races that we found in UK, but we expect there will be a lot more variation of this pathogen in different countries which has this disease, and we are now working towards understanding the diversity of these races at the genetic level, and then selecting more races in the future for phenotyping with this panel and the RenSeq data. So coming back to the disease resistance, when there are not many resistance genes known for downy mildew resistance, so it's like a bit of blank canvas when I started a year ago. However, the development of genomic resources in pea like RenSeq has recently helped to gather the pace. And we have started filling this blank canvas with some interesting resistant loci. And I hope we will continue and find resistant loci for other diseases as well. Particularly root rot is an important disease that we are working on. So with this, I would like to acknowledge people in my group. So we call our group as P-Apple. So you can see the pods. P-Pod has the people in my group, and also the group Claire Dominic's group and Noel Ellis, who really helped to start this work, and also the GIC computing team who helped us in design of the bait library, and the team of Jane at NIA with their help in phenotyping, and our funding agencies from PGRO, BBSRC, and John Ennis Foundation. So with this, I would like to stop, and I'm happy to take the questions.
Thank you, Sanu. That was really great. It's fascinating what you're doing now in peas. It's definitely some really cool work. So I will give some time for the audience to pop in some questions to that question portal. So I'll start. There's one there that I'll start with. From S. Kumari would like to know what is the true cost of the RenSeq experiment starting from the target enrichment stage? And I know that's kind of a tough question for you to answer.
Maybe you can answer this question better, Alison.
Yeah, no, yeah. Unfortunately, well, fortunately, it's one of those complicated questions to answer because obviously it varies greatly depending on the species, how large is the assay, how many samples you have. I would say in general, and maybe you can comment a little on this, Sanu, as well, the actual enrichment reagents are typically the smallest cost per sample of the experiment, because for target enrichment, you're building your NGS library first, and that's typically the sort of highest cost per sample for some people, depending on how you're building those libraries. Once you have that library made per sample, you can take it forward to an enrichment experiment or do anything else with it that you want. So you've got it, that's an immortal library now. But the enrichment reagents are typically adding some cost on top of that, but the real savings comes from the reduction in sequencing. So I suppose that would be, do you have any comments there on to what degree you were able to, you would estimate that you saved in sequencing and bioinformatic efforts by doing an enrichment approach versus, for example, whole genome sequencing for these type of samples, or is the latter not really even feasible for your wheat or peas?
Well, I mean, whole genome is definitely more expensive than RenSeq, and like you were saying, the major cost is first to design the library, and that's not only cost, it's time as well.
Oh, yes.
I think once you have library, like we only sequenced 230 lines. So if we have to sequence another 200 lines, because we have this big panel, and initially we chose this number based on, of course, the funding, and also we wanted to check whether this approach works in this panel or not. So now we know it works, and there are some resistances which are unexplained, which could be that they are rare resistances, and maybe if we increase the size of the panel, we would be able to get hold of those resistance genes as well. So I think now it is, once you have the bait library and a panel working, it's easy decision if you want to expand the size of your panel, and I think it will be cost effective, and it will take less time as well.
Mm-hmm. I know you mentioned that you used for this, either the first or maybe the second iteration, apologies, I might have missed it, some P relative species. I'm not familiar with those. Can you comment on, are those closely related species, more distantly related, and what about them did you choose?
Some of them are distantly related, and we didn't pick them actually if they were too distantly related. And we picked some of them which fall close in the phylogeny, and those which are reported in the literature are the ones which you can cross between the species easily. So we included that in the analysis, but there are some that we didn't add when we were doing the RenSeq sequencing, and I think there are some lines like fulvum, some lines of fulvum which we see are very diverse, and we would probably may not add them in the future because that might deviate the allele frequencies of the panel and can interfere with the association genetics.
I see. Okay, that's great. Is there a particular, well, let me stop there because there's a few more questions and I think I'm hogging it a bit. So second question coming from D. Botero is asking, did you find some explanation at the R gene level for the resistance in RACE11?
Sorry, but can you please repeat the question?
Did you find some explanation at the R gene level for RACE11 resistance?
RACE11. So we haven't really picked RACE11 in our analysis.
Okay. I may be misreading that question. Sorry. If you'd like to re-ask the question to clarify, that would be great. I'll move on to the next one. Will this approach be used for APR genes?
Well, that's a difficult question because most of the APR genes, they are known to be non-NLR, and we haven't done work at the adult plant stage to analyze this panel for our genes, but based on my work on wheat, most of the APR genes are reported to be non-NLR. But one interesting thing I would like to report here, which is if your non-NLR gene is an LD with the NLR genes, as you have seen in the distribution that I showed on the chromosomes for the NLR genes, so they are distributed across all the 7 chromosomes. So sometimes what has happened, it's your NLR gene is an LD with, let's say, if you have kinase involved in APR resistance, even then I have seen cases, particularly back in my work in wheat, where you get a very clear peak associated. So what our advice is, so once you get a very clear peak associated, because your NLR is an LD with that gene X, so you will see a nice peak, but always take your peaks with caution. So what we do is when we get these peaks, we analyze the complete LD region, or we at least analyze, if you don't know the LD of your crop, then you can analyze at least few kbs on both sides to see what genes are there and try to look for functional validation of those genes as well. Or maybe do some more bioinformatic analysis to see if one of them make better sense. If you can't do bioinformatics because you're restricted with NLR's genes here, so but in the functional validation, it involve add those genes, but because it could be a non-NLR. But yes, you can sometimes get as clear signal for a non-NLR genes because they are in LD with the NLR candidate.
Right. I'm sure having the best possible genome quality there certainly helps.
Oh yes, definitely, definitely.
Okay, that was all the audience questions that we had for right now, and where that works out because we'll also do to move over to Ingo's presentation. So thank you so much, Dr. Arora, for that. If you are able to stay on the line and anyone has any more questions for you, they could pop them in the chat. And if you're not able to stay on the line, I'll make sure that those get to you after the talks.
Sure. Thanks, Alison.
Of course. All right, so let me just, okay, so, sorry, too many panels. So next up, we have Dr. Ingo Hein, who will be talking to us today about his research on pathogen disease resistance in potato using the RenSeq approach. Once again, please do use that question panel if you've got any questions for Dr. Hein, and I will just switch over right now so he can share his screen.
Okay. Keeping my fingers crossed. Can you see the presentation?
I can. Looks great. We see your talk.
Fantastic. Well, Alison, thank you very much for the very kind invitation and the opportunity to present the work that we do on potato in this forum. As the title suggests, we've actually developed RenSeq into different aspects now. We cover the spectrum of fundamental research, but also we do a lot of very, very applied research. And we're very well placed for this at the James Hutton Institute. So we're on the east coast of Scotland. And you see on the image here, we're just near the River Tay. And we have a thriving potato research community. We have very close links with breeders, with farmers, with the industry in general. And we also have a good research base, and we have a fantastic resource, which is the Commonwealth's potato collection. So that's the source of our resistances and where we're looking for new traits, basically. I'd also like to use this opportunity to really introduce some of the very talented students and members in our group. So most of the work that I present actually comes from our students and the postdocs within the group. Now, to set the scene, potato is the most important non-cereal food crop. It's the third most important crop in the world. More than 1 billion people consume potato, and annually we produce over 300 million metric tons of potato. When we think about diseases, quite often late blight comes to mind, and you know it's a disease that caused the famine in the west coast of Scotland and in Ireland in the 1840s, and it has really dire impact on the entire population of Ireland and the west coast of Scotland. More than 1 million people died of starvation, and more than 2 million people had to emigrate. And actually, that's one of the facts why we see so many Scottish and Irish scattered all over the world. In fact, the population of Ireland has never really recovered from the impact of famine. So what happened? A pathogen, an oomycete pathogen called Phytophthora infestans, which translates into plant destroyer, came into Europe, made its way into the UK, and literally we had subsequent crop losses for a number of years in potato. So it is still one of the most important diseases we are working on, but there are other diseases as well that are of importance to the industry. One of these, for example, is PCN, potato cyst nematodes. And the moment you have PCN in your field, you cannot use your field for seed potato production. And the Scottish seed potato industry in particular is very, very keen on keeping PCN out of the fields as much as they can. So looking at our production, we are producing more than 300 million metric tons a year, but we are losing about 25 to 30% of our crops to diseases. And that's something we obviously want to address. And that's where RenSeq comes in its own. So we are interested in the functional nucleotide binding leucine-rich repeat, or the NLR class of resistance genes, to control these diverse pathogens of potato. And we know that NLRs are involved in controlling viruses, nematodes, especially PCNs. We also use it to study filamentous fungus diseases and against oomycete, so late blight disease. So to sort of set the scene where we started off, we had a joint PhD student, Florian Juppe, with the group of Jonathan Jones at the Sainsbury Laboratory. And we basically took the potato genome that was released in 2011. We've done an annotation on the disease resistance genes, on the NLRs, and then we developed the first RenSeq library to study these resistance genes. Now, we didn't know how well this technology would work. So as a proof of concept, we actually applied this targeted enrichment sequencing on the sequenced potato clone, which was a doubled monoploid called DM, just to basically see, do we find all the genes back that we designed baits for? And the answer is yes, we do, but we actually identified over 350 additional resistance genes. So we could actually use the technology to pull out all the resistance genes that were missed on the computational annotation of DM. And what we've learned from this exercise was you don't have to have a 100% match between your bait and your target. About 80% sequence identity is sufficient to facilitate this enrichment sequencing. And we've been using RenSeq ever since to, for example, map resistances against different pathogens. We've also developed a different analysis. We call it diagnostic RenSeq or D-RenSeq. And that actually has the advantage that it allows us to look for known functional resistance genes, either in wild species or in established cultivars. And that's something breeders are very, very keen on. We use this, for example, to inform breeders which cultivars they can use in their breeding programs to combine complementary resistances. But we can also develop very informative and highly transferable markers for these resistance genes. And then, as Sanu introduced so beautifully, we're also using association genetics RenSeq, RenSeq, where we basically use a KEMA-based association approach. So I'd like to start off by introducing our D-RenSeq approach and how we're using this to identify new resistances in wild potato accessions. So for diagnostic RenSeq, what we tend to do, we take a number of potato plants, they extract the DNA, we index the DNA, we conduct the target enrichment sequencing, and then we take our sequence reads and we ask the questions, well, do we have representation of any of the known functional resistance genes within our reads? So each of these square boxes that you can see here on the screen represents a known functional resistance gene. For example, here we have a nematode resistance gene called GPA2, and you have the start and the stop of the gene on the x-axis, and on the y-axis you have the coverage that we achieve with our RenSeq reads on a log scale. So we're having 10 reads or 100 reads here. So basically what we're doing, we're mapping the RenSeq derived reads, Illumina reads, back to these reference genes with real high stringent conditions. We're not allowing for any mismatch rates, and we're looking for full representation of these genes. And in this particular example, we looked at 6 potato cultivars, while 3 of them are transgenic lines. They're called acclimate, glaciate, and hibernate. And we can basically confirm that the transgenic plants contain the resistance genes RPIV and T1.1. None of the progenitors, so Ranger Russet, Russet Burbank, or Atlantic contains these genes. We can also show the difference between RPIV and T1.1 and RPIV and T1.3. You can clearly see where we have sequence polymorphisms that distinguish these genes. And what we could also show is that Atlantic, and thereby hibernate, the transgenic plant derived from Atlantic, also contains the nematode resistance gene GPA2, the late blight resistance gene R1, and the virus gene Rx. So we see presence-absence variations. We also see partial coverage, for example, here for R2, but we conclude that this gene is not present and not functional because there's a huge chunk of the gene missing. So we're looking at the Commonwealth potato collection to identify novel resistances against diverse pathogens. So the Commonwealth potato collection, or the CPC, was actually established in the 1930s with collecting expeditions in Mexico and South America. And these sort of collections took place as a consequence of the farming. It was a realization eventually that you can use wild species to integrate resistances into cultivars. And we're not the only country in the world that has a potato germplasm collection. There are others, probably, well, many of them are actually bigger, such as the collection at the International Potato Center, for example, or our colleagues in Wageningen or in Sturgeon Bay in the US. So there are many of these collections throughout the world. Our collection has over 1,500 accessions. We have over 80 species represented. Two-thirds of them are wild species, but 1/3 of them are cultivated land races. So they're not too far away in terms of breeding to make cultivars out of them eventually. Now, the CPC is maintained as true seeds and is on a very slow cycle of rotation. There's a 20-year cycle, and that's done deliberate because every time you grow up seeds from this CPC, you actually lose some of the diversity. And here's a typical snapshot from a glasshouse at our institute. There's about 60 to 80 accessions are grown every year, and we have multiple replicates per plant, so that they come from true seeds. And we cross between these to try and keep as much of the diversity as we can. So what we have done so far is we've done Illumina-based RenSeq of 150 diploid accessions from the CPC, and also with colleagues at the International Potato Center in Peru and also in Asia, we've actually sequenced additional accessions for them. We've also conducted PacBio RenSeq. So far, just shy of 100 diploid accessions from the CPC. Our aim there is to describe the pan-annularium of potatoes. We just got the data, so we're very excited to look into this one now. And we've really sort of phenotyped these accessions very well with diverse late blight isolates last year, and we are starting PVY and nematode screens actually this year. We also have historic data that we can draw on for some of these accessions. The main driving questions that we have is, can we identify new resistances? So how do we go about this? We start off with a phenotype. We have a resistant plant. And at this stage, we don't know anything about this plant. What we want to avoid is a lengthy genetic study that actually then points to the presence of a known resistance gene. So what we're doing at the immediate onset of our study, we do a diagnostic RenSeq study, and that gives us a binary answer. Either the resistance is based on a previously characterized gene, in which case we are no longer interested in this plant, or we have a novel resistance. If we have a novel resistance, we then typically set up a cross with susceptible plants to generate a segregating population. And then we use a bulk segregant RenSeq-based mapping analysis, for which we actually only need the resistant bulks and susceptible bulks and the susceptible parent, because we already sequenced the resistant parent at this stage here. So it's actually highly cost-effective for us as well. And the key really is we're not going after resistances that have already been described in the literature. So we have a very easy way of eliminating those. Here's an example where we screened 12 accessions of Solanum bulbocastanum from the CPC. We did drop inoculations, and basically when you get a strong hypersensitive response, you actually see the cell death on these leaves. And out of these 12 accessions, 11 were resistant, one of them was susceptible. After we've done our RenSeq and the d-RenSeq analysis, we actually concluded that only 2 of them have a novel resistance gene. For all the others, we could actually show that the resistance is based on the presence of BLB1 or a BLB1-like gene. We've seen a number of these BLB1-like genes, all of which we've actually then cloned and tested functionally. They were functional. And we ended up with these 2 accessions here that have a resistance that we cannot explain by the presence of either BLB1, BLB2, or any other known functional resistance gene. And that's when we then did our genetic study. We did our cross. In this case, we did a cross with Solanum microarcanum, and we got a population that segregated 1-to-1. So similar to Sanu's slide here, what I'm showing you is the distribution of resistance genes on the 12 chromosomes of potato. So you see every chromosome basically has a resistance gene, has a number of resistance genes. And you clearly see these big clusters here. For example, we have the R2 cluster on chromosome 4, and we have the R3 cluster on chromosome 11. When we did our bulk segregant analysis and we filtered for SNPs that are linked to our resistance phenotype, we could actually map this resistance to potato chromosome 5. And I think that is really good news, because we knew from the other resistances that came from Solanum bulbocastanum, so there's a resistance gene BLB1, BLB2, and BLB3, they actually map to chromosome 8, 6, and 4 respectively. So we now know for sure we have a new resistance from Solanum bulbocastanum. It maps to chromosome 5. No other resistance has ever been mapped there, as far as we know, from Solanum bulbocastanum. So moving on, using our d-RenSeq approach in cultivars. Now, we've done RenSeq now for over 270 varieties using Illumina sequencing. We've also done just shy of 100 breeding clones for the commercial arm, for example, from the James Hutton Institute called James Hutton Limited, but also for other companies. And we've also done some PacBio sequencing. And I'm coming back to the significance of the PacBio sequencing at the last part of this presentation, association genetics. Now, first of all, we could use our d-RenSeq data to look at the historical pattern of resistance gene deployment in cultivars that are grown in the UK. And with the help of Sasa in Edinburgh, we could actually get material that was grown during or prior to the farming years. So the cultivar Lampa, for example, that was the #1 cultivar grown in Ireland and the west coast of Scotland, because it gives very, very good yield on marginal land. And it was bred around 1800 there. And of course, our d-RenSeq analysis indicated it doesn't have any functional resistance gene. And the same is true for all of these other genes that we cultivars that we sequenced. After the farming, there was a realization that breeding had to kick in to look for resistances. And the first thing the breeders did, they looked for seedling selection. So no new genes were introduced. There was no crossing going on with wild species. They just looked for different alleles in existing materials. And all the cultivars that are derived from this period, including some that you might recognize, such as Russet Burbank or King Edward, they're still grown today and consumed today. Again, they don't contain any known functional resistance gene and are subsequently completely susceptible to most diseases. And then, as I mentioned early on, the realization came that we can use wild species to integrate resistances, and we see this now after the 1930s. So we see in the 1940s here, we have the cultivar Craig Snow White that contains the late blight resistant gene R1. We see others that contain R1 or the R2 family or R3A and R3B. We also see cultivars that have GPA2 and RX being introduced in them. And then with the availability of good genetic markers and cloning becoming easier, we actually see other resistances such as BLB2 and VNT1 coming in cultivars. So there is a trend that we see new resistances coming through. However, if we look at the top 50 potato cultivars that are grown, they actually fall, unfortunately, into this category. So that's about 80% of the land used in the UK to grow potatoes have either no resistance gene or they have been recycling the same resistance gene over and over again, all of which have been overcome now by variation of the pathogen. So they don't provide any resistance at the moment. So we've moved on from looking at the historical deployment of resistance gene to really sort of get a better understanding how resistance genes are used throughout the world. We have a BBSRC project that has allowed us to look at a number of cultivars grown in Europe, but also in the US and of course in the UK. Through a GCRF funded project, we had the opportunity to work with the International Potato Center, so we could study varieties or breeding clones from South America, Africa, and also from Asia. And really that has given us an unprecedented insight into the deployment of resistances. I sort of summarize it very briefly here. In red, we've shown you the varieties. So we looked at over 270 varieties, and in blue, we have breeding clones. And if we look at the already defeated resistance genes, we can tell you they are in high abundance in a lot of the cultivars that are currently being grown, but also that are already being bred at the moment. In terms of resistance genes that do provide protection either against late blight, such as R8 or VNT1, BLB1 or BLB2, we start seeing them coming through in breeding clones, but not at a scale that we were hoping for. And we also see sort of the RY store resistant gene effective against PVY being in some of the cultivars or in some of the breeding clones, but again, at a very, very low level. So this is an opportunity for us now. We've shared this information with our breeders. We made them aware of cultivars that have complementary sources of resistances. And the breeders have actually set up breeding programs to combine these resistances, not just late blight, but also the PVY resistance and others. So I think we're going in the right direction there. But working with breeders, one thing I've realized is they are very fickle. They're very, very difficult to keep happy. So they always want more. And what they've asked us for is, we now know which varieties to use for combining resistances. Can you also give us markers? So we looked into this. And again, we used our RenSeq approach for this. So in this panel here, you see the resistance gene R8 effective against late blight. And I show you two cultivars here that have R8. So again, you have the start of the gene, you have the end of the gene, and you get full coverage of our RenSeq reads. Now, based on this information alone, it'd be impossible to design a marker that actually works, that is transferable and informative. However, the picture changes the moment we start looking at cultivars that are susceptible to late blight and do not have R8. The moment we bring the non-functional genes into the picture, you actually start seeing any primer designed for these areas here that are very conserved, irrespective if you have a functional or a non-functional gene there. These would give you false positives. They would not make good markers. And you actually realize, if you sort of compare the non-functional gene to the functional gene on a larger scale for many, many cultivars, there are actually very, very few places left where you can design markers that inform breeding. And that's exactly what we've done. We've homed into these regions that are specific for the functional gene. They're not present in the non-functional genes, and we converted this into a Cas marker. So these Cas markers are very powerful markers. They're allele-specific markers. So we can assess if we have the absence of the gene, so we have a non-functional gene present, for example, but we can also inform the breeders if they have a single copy or two copies, if it's a duplex combination of a functional resistance gene. And we've developed these markers now for a number of important resistances against late blights, such as R8, R9A, BLB2, and BNT1, but also for the RY store resistance genes, and we developed them for nematode resistances, such as H1 and GPA5. And we're going to share with you some data that we have generated on GPA5 quite recently. So that brings us to the association genetic part, where we literally take advantage of the existing data that we have already. And thanks to Sanu's really great introduction, I don't have to go on about the slide. It shows how the association genetics works. Very shortly, you have a representation of a cultivar in our case with these contexts down here. And then you use k-mers, so subreads from cultivars with different or contrasting phenotypes to look for association between the context and the presence of these k-mers. So the higher the peak, the stronger the association, the broader the peak, the more k-mers you have associated with this particular phenotype or this context. So as a proof of concept, we wanted to find R2-like in the cultivar Innovator. Now, we knew that Innovator contains an R2-like gene. We established this with our RenSeq pipeline, and we used an Illumina-based NLR representation. We took a combination of varieties and breeding clones. So in total, we looked at over 330 accessions or varieties that we combined. Of those, 43 contained R2 according to our RenSeq data, and 291 did not contain R2. So when we did our association study, so on the x-axis, you have the number of resistance genes that have been assembled based on the Illumina read of the cultivar Innovator, so just over 2,000. And then you have the association on the y-axis, and we basically said everything above this line here, a score of 30, is positively associated. We ended up with 16 contexts that are positively associated for R2 or R2-like. When we looked for the closest homologue of R2, we were actually somewhat disappointed, because although we found something that has identity to R2, it's actually only 50% of the entire R2 gene. So using Illumina reads didn't help us to identify the full-length gene. So we changed our approach, and we took PacBio reads to represent the cultivar Innovator, or the resistance gene of Innovator. And again, if you look at the graph, we now have slightly more resistance genes, just shy of 3,000 NBS-LRs that we predict in Innovator. But we could find all our benchmarking genes. We knew, in addition to R2, there was also R1 and R3A and R3B present in Innovator. All of them were appropriately assembled in the PacBio data. When we did our association study using exactly the same condition we did before, we identified 18 contexts that are associated with R2. And as I mentioned before, we actually now find full-length R2 in our association panel as well. And that comes out positive. Interestingly, the other peaks that we've observed so far, when we have a closer look there, they're actually pretty much all R2-like genes. And that reflects that R2 is a very, very big cluster. It's the biggest cluster we have in potato. So the association study works beautifully to the point now that we can identify full-length genes, even if we look at the most complex family. So we applied this to look for nematode resistances, and that's something where the Scottish government is actually very keen to identify at least markers for these resistances. And to identify marker, we have to get an idea which genes could be underpinning these resistances. So in terms of potatoes as nematodes, we have two different species. We have Clovidera rostokiensis and we have Clovidera pallida. Rostokiensis is known as a golden nematode and pallida is known as a white nematode. And we We're somewhat lucky in the UK. We tend to have only the pathotype RO1 in the UK, so it looks like it was a single introduction that took place. And actually, that's controlled by a resistance gene called H1. Now, H1 was discovered in the CPC in a species called Solanum tuberosum, subspecies Andigena. That was accession 1673, and that was actually identified in the 1950s. The gene has been used in a lot of cultivars now, but it's still elusive. So nobody has been able to clone the H1 gene up to date. We are now in a place where we have 4, maybe 5 very strong candidates for H1 that we are trying to analyze functionally, again, with our colleagues also at the Sainsbury laboratory. For Clobidera pallida, there's a gene called GPA5, and that controls the pathotypes PA2, 3, and 1. And the resistance is derived from Solanum vernii, and it was first deployed in the cultivar innovator that I introduced earlier on. And it's also known as the innovator resistance. So we are now trying to identify candidates for GPA5 too. So again, we used our PacBio-derived representation of the resistance gene from innovator. We had 128 potato cultivars with contrasting phenotypes, 24 that had the GPA5 gene, or at least the resistance phenotype. Some of them are actually breeding clones, not varieties. And then we had 104 varieties that are susceptible to Clobidera pallida. When we did our association study, again, we got positive association. We had 14 contexts that were positively associated. These contexts are very big because they're PacBio-based contexts. They can actually have multiple NLRs. So we actually have more than 14 NLRs that made it through the analysis. But I give it away now, we actually have 9 candidates that are remaining. When we map these 14 contexts to the chromosomes of potato, again, you see here the bars showing the representation of the different resistance gene loci. You actually see that we have very strong association here on potato chromosome 5. So these clades here map within a very, very small interval of chromosome 5. And actually, that's the correct position for GPA5 because we know it should map to the top end of chromosome 5. We have 2 outliers. One of them has a very, very poor score, a very poor KMA representation. So only 5 KMA suggests that this one is a strong candidate, and we can dismiss it based on this. The one interesting gene on chromosome 7 has very good association. Lots of KMAs are associated with it. It's probably on the wrong chromosome, but interestingly enough, this particular resistance gene in this context is actually over 90% similar to the gene in this context 6.1. So we actually think it's probably just been mapped to the wrong chromosome, but it looks fantastic in terms of data. Now, how do we know that we have strong association? And that's where we're using the RenSeq again. So we've taken all our candidates. We had 16 candidates that came into the closer selection based on these 14 associated, or these 12 associated contexts, I should say. And what we then did is we took these candidates, we put them in our RenSeq pipeline, and we basically using graphical genotyping here to represent us. Cultivars that have GPA5, shown here in green, they typically have 100% coverage of these candidates. Cultivars that do not have GPA5 and that are susceptible, they typically have very little coverage of these candidate genes. And what is interesting is we look at these 16 genes here, we can rule out the bulk of the genes on this side here because of a beautiful recombination event here that basically rules them out. So this particular clone that we've selected here doesn't have the presence of the resistance genes 10 till 16, but the phenotype is still resistant. And we see other recombination events and other genes as well. So we are very confident that our best association is with these 9 candidates. And we're now taking them forward for functional studies. So in summary, with RenSeq, it allows us to identify resistances from the Commonwealth's potato collection, but also other germplasm collection, and we're working with collaborators throughout the world. We can track functional resistance genes in cultivars and breeding clones. We can help breeders with the selection of good parental material for crosses. We can also provide markers for our breeders so they can track these resistances in segregating populations. And using association genetics, we can actually identify candidate genes for resistances that are already deployed in cultivars, but which remain elusive. And as I said early on, science is very international. We work with a lot of organizations throughout the world. And, you know, I'd really like to thank all my colleagues at the Sainsbury Laboratory in Wageningen, Sasa in Edinburgh, in Kars in Beijing, colleagues at SIP, Yunnan Academy of Agriculture Sciences, and also China Agricultural University. And like to thank the sponsors of our research, the Royal Society, Innovate UK, BBSRC, the Scottish Government, and the companies Simplot, McCain, Agrico, and Greenvale. And thanks for your attention. Oh.
Well, thank you so much, Ingo. That was great. I have a couple questions here, so I'll just jump into those right away. So potato, it's a relatively small genome. I guess as far as crops go, right? It's less than 1 gig, as I understand it. So, you know, for those, it seems that maybe a non-enrichment strategy would be potentially an option as well. Is there definitely still an advantage for you guys doing the enrichment to get that high coverage for those NLRs? Or is there...
Yeah, I think it's a great question, and you're absolutely right. The potato genome, you know, compared to other genomes, is relatively small. It's 840 million base pairs. And actually, we did a comparison. We had the exact same question: is it worthwhile doing? The advantage we get with RenSeq is that we have really good coverage of the genes that we are interested in, either being a known functional resistance gene, or if we look for new resistances, we know they are properly represented. So if we do the same approach with non-enriched data to get the same information back, you actually need 88 times the information that we would get from our enrichment sequencing. So you actually have to spend the equivalent, not money, but you need a much better sequencing depth if you don't do enrichment sequencing. So you need 88 times enrichment that we, without the enrichment, basically, to come to the same conclusion. So based on this, and also the sequencing costs are coming down, it is still worth our while to do the target enrichment sequencing, just so we know reliably we have the coverage that we need for all these resistance genes.
Great, thank you. Is there a particular read coverage that is being aimed for in these studies? I think 100 was on one of those figures.
So we try and get our read coverage between 10 and 100 for these genes. I know it's a huge interval, but there's so many different factors that also impact there, and it looks like the quality of the DNA is one of them that has a huge impact on how the enrichment sequencing works. So you need a minimum of 10, and we're trying to aim for between 30 and 50 ideally, but we tend to end up between 10 and 100 on average.
And that's for both Illumina and for the PacBio work?
PacBio is less, so we're looking for about 30 to 40x coverage typically. It's also much more expensive.
Right. Okay. Are you aware of RenSeq being used directly in any commercial research programs, or because you mentioned the work with the farmers to develop the CASP markers? Is RenSeq used in any other research programs that you're aware of?
Yeah, so our own institute, so the James Hutton Limited is a commercial arm of the James Hutton Institute. They use the data from our RenSeq approach. Basically, they breed for some of the big international companies. So yeah, it really sort of filters into the production of potato cultivars. We don't do it exclusively for them, so we work with other companies as well. Some companies have sent us samples and asked, can you have a look what's in there? Some of the breeders have sent us material, again, asking the same question, can you help us identifying the source of this resistance? We are open for collaborations, and we are really happy to work with our colleagues and breeders. So yeah, we don't do it exclusively for James Hutton Limited.
Great, that's great to know. Okay, from Simon M asks, both speakers today have talked about specific R genes being present only in resistant genotypes. Is presence absence the norm for our genes, or do coding sequence variants and differential expression confer resistance too?
So we do see a presence absence variation. There are also a lot of variations within the coding sequence, even within functional genes. We see a lot of variation in genes that have been published where we find polymorphisms. They have retained their function. Some of them we might actually, well, we're trying to do the research to show that they actually have a gain of function with these polymorphisms as well. But overall, when we look, especially in cultivars, we tend to find relatively high representation of R8-like genes, for example, even in cultivars that do not have R8. It's not like complete presence absence. We still see between 40 and 60 or 70% coverage of parts of the gene. And that's actually one of the aspects why it is so difficult to develop markers for these resistance genes, because it's not just a simple presence absence in cultivars by the looks of it.
Are the markers that you developed, this is just a me question, are the markers that you've developed, those CASPA markers, they're typically within the actual NLR sequence, right? Not like adjacent? Okay. And that is possible even though NLRs they have these relatively conserved regions, because you have such a good knowledge of the diversity of NLR sequences that you're able to develop markers even for a very specific allele.
Yeah, that's exactly right. It's the ability to contrast a functional gene from all the other non-functional genes that are in our material. And potato is a tetraploid, so every cultivar we look in, we potentially look at 4 alleles as well.
Are you typically resolving the sequences of those alleles in the haplotypes, or not necessarily?
Only with PacBio data. I would not want to try this with Illumina data. It's incredibly difficult. And to generate markers, we don't have to. We just have to see where are regions that are unique for the functional gene that are not present in any of the other cultivars that are susceptible, for example.
Great. I believe that is the end of the questions. I apologize to anyone if I missed one in the panel. So with that, thank you so much, Ingo. This was great. You're still looking at my screen, right? So I'll just wrap us up today by saying again, thank you so much to both of our speakers and to everyone who tuned in today, and for all those questions that you submitted. That was extremely informative. Both Dr. Arora and Dr. Hein have graciously agreed to allow us to post this session later online. So if you would like to catch the recording again, you can do that as well later. It'll be available on the Dysel Arbor Biosciences website. We'll also have their email addresses here as well if you would like to reach out to either of them directly regarding their research programs. And of course, the Arbor contact info is there as well if you have any questions for us directly about utilizing the reagents that we sell for RenSeq in your research, either in your own lab or a project through our full service NGS lab. So thank you very much again to both of our speakers, for you all for being here with us today, and wishing everybody a pleasant rest of your day. Thank you.
Thank you. Bye.